TIMER consists of three major components: immune, exploration, and estimation. The immune component contains four modules that allow users to investigate the association between estimated immune infiltrates and gene expression, somatic mutations, somatic copy number alterations, and clinical outcomes in the TCGA cohorts. The exploration component has four modules that allow users to find non-immune related associations in TCGA. Users can compare the expression of a gene between tumor and normal and find the associations between the expression of one gene with patient survival or the mutation status of an expression level of other genes. The estimation component can infer immune cell infiltration of user-provided expression profiles using the 6 estimation algorithms. Instruction for each module is provided on the module page. The relationship between tumor gene expression and immune infiltration is of great interest to many researchers. The gene module allows a user to identify such relationships in a fast, comprehensive, and unbiased way. If a user selects a gene in an immune cell type, TIMER generates a heat map table of the Spearman correlations between the expression of the input gene and the abundance of the immune cell type, as well as the subtypes in a 59-cell hierarchy across all cancer types. Red and blue represent positive and negative associations with statistical significance, respectively, and gray represents non-significant correlations. This display allows the user to have a better sense of how robustly expression of their input gene is associated with the immune cell type across different cancer types using different estimation algorithms. When purity adjusted option is selected, clicking on any of the numbers in the table will result in two scatter plots showing 1. The correlation of the gene expression with tumor purity, the proportion of cancer cells in a sample estimated by CHAD, and 2. The association of the gene expression with the immune cell type. It is worth noting that tumor purity is a major confounding factor in this analysis, since most immune cell types are negatively correlated with tumor purity. Therefore, we recommend users select a purity-adjusted option, which will use the partial Spearman's correlation to perform this association analysis. When purity adjusted is deselected, we see that we only get one plot. Somatic mutations play crucial roles in tumor regenesis and profoundly influence patient treatment response and survival. Researchers are often interested in the effect of gene mutations on tumor immune environment, specifically immune cell infiltration changes. The mutation module allows users to analyze and visualize the effect of gene mutations on immune cell infiltration across multiple cancer types and immune cells types simultaneously. A user can input a gene by either manually typing the gene name or selecting from a list. Given the input gene, TIMER displays a bar plot showing mutation frequency of the gene for each TCGA cancer type. When the user specifies the immune cell type and clicks Submit, TIMER presents a heat map table of log-fold changes of immune infiltration level between tumors with the input gene mutated and tumors without. Red and blue represent statistically significant higher and lower infiltration levels when the gene is mutated, respectively, and gray represents non-significant changes. Similar to the previous module, the user can click a spot on the heat map and see violin plots of immune infiltration distribution in the mutant versus wild-type tumors. Genes with somatic copy number alterations are hallmarks of tumorigenesis and progression and could influence immunotherapy response. The scNA module allows a user to compare immune infiltration distribution by the scNA status of a gene across TCGA cancer types. GISTIC 2.0 estimates the scNA information from copy number segmentation profiles at the gene level, including deep deletion, arm level deletion, diploid normal, arm level gain, and high amplification. Given the input gene, TIMER displays a stacked bar plot showing the relative proportion of different scNA states of the genes for all TCGA cancer types. TIMER requires the user to specify deep deletion or high amplification alteration status of the genes to compare with the diploid normal status. When a user submits a query, TIMER draws a heat map table for the log-fold changes of immune infiltration levels between the specified alteration group and the normal one. Red and blue denote a higher and lower immune infiltration with statistical significance, respectively, and gray denotes non-significant changes. With a click on an entry of the table, the user can view the immune infiltration distribution between different scNA status of the gene on a violin plot for pairwise comparisons of normal group with each alteration group. Tumor-infiltrating T cells and B cells have been reported to affect patient clinical outcome. The clinical relevance of the immune infiltrates can be learned by examining associations between immune infiltration level and patient clinical outcome. The outcome module allows a user to quickly evaluate the association of immune subset abundance in patient survival across TCGA cancer types. TIMER uses Cox proportional hazard model to evaluate the effects of immune infiltration level, specific gene expression, and patient clinical information on the hazard ratio. User input variables, except the immune cell type, are considered as confounding factors for adjustment of the regression model. Once a user submits a query, TIMER generates a heat map table of normalized coefficients of immune cell types. Each spot on the heat map table stands for an independent Cox model, which only includes one immune cell type and the confounding factors for one cancer type. Red and blue represent significantly decreased and increased survival respectively, and gray denotes non-significant associations. The user can click on a spot to examine how the immune cell and gene expression affect patient survival on Kaplan-Meier curves. One may also customize the cutoffs for immune infiltration and gene expression to stratify patients. To evaluate whether a gene has therapeutic target potential, the gene often needs to be uniquely expressed or more highly expressed in tumors than most normal tissues. The Gene Differential Expression module allows users to rapidly compare a gene's expression level between tumor and matched normal tissues across all TCGA cancer types. When a user inputs a gene, TIMER displays box plots of the gene expression distribution in tumors versus normal tissues across all cancer types. The statistical significance computed by the Wilcoxon test is annotated by the number of stars. Gene expression-based approaches have been developed to predict survival and therapy response. The Gene Outcome module allows a user to simultaneously evaluate the gene expression association of multiple genes on patient survival across TCGA cancer types. Similar to the Outcome module, TIMER utilizes the Cox proportional hazard model for association evaluation. Variables belonging to clinical or histological features such as patient age, tumor stage, and purity are considered as confounding factors in the regression model. Once a user submits the request, Timer draws a heat map of the normalized coefficients of the input gene expression in the Cox model. Each spot on the heat map represents an independent Cox regression coefficient, which associates the single gene expression and other covariants to the hazard ratio for the corresponding cancer type. Decreased and increased survival with statistical significance are denoted by red and blue respectively. A non-significant association is shown in gray. The user can click a spot on the heat map to investigate the association between the gene expression and survival in the Kaplan-Meier curves. In this figure, Timer initially divides samples into high and low groups by the median of gene expression. The user can customize this division cutoff as well, and the corresponding hazard ratio and log-rank p-value are simultaneously updated on the Kaplan-Meier plot. Identifying the functional impact of somatic mutations in the gene expression pattern is critical for precision oncology and drug discovery. The gene mutation module allows a user to assess how the mutation status of one gene influences the expression of the mutated gene and other genes across TCGA cancer types. Similar to the mutation module and immune component, when a user selects a gene with recurrent mutations in TCGA, Timer shows a bar plot of the mutation frequency on this gene in each TCGA cancer type. When a user enters both recurrently mutated genes and genes for the expression query, Timer displays a heat map of log-fold changes of gene expression between tumors with the gene mutation and tumors without the gene mutation. Red and blue represent statistically significant higher and lower gene expression levels when the gene mutation is preserved, respectively, and gray represents non-significant changes. The user can click a spot on the table to view detailed violin plots of gene expression distribution in mutant versus wild-type tumors. The gene correlation module allows a user to discover the co-expression pattern of genes across TCGA cancer types. Given one initial gene of interest and up to 20 other genes, Timer generates a heat map table of Spearman correlation of gene expression between the gene of interest and other input genes. Red and blue indicate positive and negative associations with statistical significance, respectively, and gray indicates non-significant associations. The user can click a spot on the heat map to view scatter plots of the expression correlation between two genes. In the case of confounding effect from tumor purity, a user could also select purity adjusted option, which enables Timer to use partial Spearman correlation to conduct this analysis. Given the prevalence of transcriptome studies across immuno-oncology, we improved the capacity for immune infiltration estimation and interpretation of user-provided expression profiles. The estimation component accepts a gene by sample expression matrix in a CSV or TXT file formatted with standard delimiters. The values in this matrix should be TPM normalized without log transformation to fit the requirements of all estimation methods. Once the input file is uploaded, Timer automatically shows the number of samples so that the user is able to confirm that the file was uploaded correctly. TIMER adopts the immune deconv package to infer immune cell component abundance of the input gene expression data using 6 state-of-the-art algorithms listed on this page. TIMER algorithm takes tissue-specific effects into account and needs cancer type information to improve the accuracy of the estimation. If the user selects auto as the cancer type, TIMER will automatically assign the sample to the TCGA cancer type with the highest expression similarity. Once the user uploads the gene expression file and starts the run, TIMER will run immune infiltration estimation and show a progress bar. The results are summarized in a downloadable data table with the estimated values of all immune infiltrates for each sample from each algorithm. It is worth noting that the estimation from different algorithms have different interpretations. While all 6 methods allow comparisons of the same cell type between samples, CyberSort, QuantiSeq, and EPIC also allow comparisons of the different cell types within the same sample. To illustrate the difference, we examine the 2 images TIMER draws when input sample size is smaller than 10. The first image is a multi-panel bar plot comparing the infiltration level between samples for 8 common immune infiltrates estimated by each method. The second image is a pie plot showing the proportion of the 8 immune infiltrates estimated by CyberSort, QuantiSeq, and EPIC.